Hello everyone, welcome back to Chinmoy's Biology channel. So today we are here back with the second part of the lesson, genetics and evolution. So in your previous class, I have already given you an introduction of heredity. We have discussed about the alleles, about the genotype, phenotype, dominant and recessive factors. So we have already spoken about all those things and we have started about the monohybrid cross. So today we will be continuing with the monohybrid cross and we will complete the part of incomplete dominance and other parts we will be studying today. So please codominance and all will be continued today. Multiple alleles also we will cover up. So don't skip in between. Stay tuned throughout the full episode, and if you have not yet gone through the previous episode, please do go through it. I'll be putting the link in the description box. So go through it and then continue with today's lesson. So without further delay, let's get started with today's lesson of genetics and evolution. So last day we have already spoken about the dominant, the recessive characters. We have spoken that T, capital T is taken for tall and dominant character, and small t is taken for dwarf character. Okay, so it is not that dwarf means D. Okay. So this is convenient to use the capital letter for the dominant one and the lowercase letter for the recessiveness, right? So now since TT is a heterozygous for genes controlling one character of height only, so it is a heterozygous gene and it is monohybrid, and the cross between TT and TT is called the monohybrid cross. So what is happening over here? See, we can see that alleles can be similar as in case of homozygotes, that is TT and TT. That is, it is fully homozygous. It is dominant is tall plant, and this is recessive, that is a dwarf plant, that is also homozygous. It is not heterozygous. Or can be dissimilar as in the case of heterozygous or TT. Okay, so hence TT plant is heterozygous for genes controlling one character height only. So in the monohybrid cross, we will be taking two homozygous characters only. So let us see the cross what we are doing over here. So we are crossing a pure tall plant with a pure dwarf plant. So this cross is happening. So when we are seeing here what is happening, these are the gametes. TT is also a gamete. So one is male one and another is female. So I don't know here that which one is male and which one is female. So we are randomly doing this. So this we are crossing. So what will be the result of that? So TT, you can see, then TT, again this one is TT. Then again we are multiplying this TT and TT. So all are heterozygous in characters, in genes they are heterozygous, and they will be all same type of plants. So what we will see that in the genotype they are 100% heterozygous, but 100% they all are tall plant. So here only the dominant factor tall plant is being visible. So 100% are tall. So the phenotype is that is 100% are only tall plant. Okay. So from the observation that the recessive parental trait is expressed without any blending in F2 generation. So in F1 generation what we are seeing that no blending is there. 100% tall plant is only there. So in F2 generation what is there? Then again these two plants TT and TT, from these F1 generation selfing is done. Then what is the result we are getting? See, now these are the gametes, TT in one side and TT in the other side. So this is the Punnett square what we are working on, right? So then we are getting, see, T and T we are multiplying, we are getting TT, we are getting the next one capital T and small t, the next one capital T, small t, and the last one is T, that is small, that is recessive. So what will be the phenotype? The phenotype is 3:1. So in F2 generation the phenotype is 3:1. But the genotype is 1:2, 2:1. So this is pure homozygous. This gene is pure homozygous tall, pure homozygous dwarf, and this is heterozygous. So 1:2, 2:2, 2:1 is the genotype. So the Punnett square used to understand a typical monohybrid cross conducted by Mendel between the true breeding tall plants and the true breeding dwarf plants. So the first filial generation, the result was 100%. In the genotype was heterozygous, all were heterozygous, the genes were, and the phenotypically all were tall. But in the F2 generation, we have found certain variations. So now, without any blending in F2 generation also, what we can infer that when the tall and the dwarf plant, these gametes are produced by the process of meiosis or cell division, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to the gamete. So, we are seeing that from one parent, one gamete is only coming to the next generation. So, they are pairing, only one gamete is pairing with one another. So, they are segregating, they are separating from each other, and only one is transmitted to the allele. So, you can see they are separated. Only one is transmitted from here and another is transmitted from here. So from either of the parents, only one gene is coming. So the segregation of alleles is random process, and so there is 50% chance of gamete containing either of the allele. And it has been verified by the results of the crossing that in this way, the pure tall plants have the allele T and the gametes of the dwarf plant TT have the allele T. So is it clear? So if it is TT, they have the gamete T, and if it is T and T, they have the gamete T. Okay. So what is happening due to fertilization of these two alleles? So T from one parent is coming through the pollen and T from the other parent is coming through the egg. So this is what and how they are united to form the zygotes. So is it clear to everyone now? So I think you were confused, thinking that what all is happening between the dominant and recessive part. There from T is coming, there for small t is coming. No, it's not like that. So the genotype is capital TT or small tt or capital T small t. So either homozygous or heterozygous. So the one of the alleles is coming from one parent through pollen and another is coming from another parent through the egg. So they are united to form the zygote, and that one T allele and one T allele, they are combining to form the zygote. So in other words, hybrids have TT. Okay, so since these hybrids contain the alleles which express contrasting characters, hence they are called heterozygous. And the production of gametes by the parents, the formation of the zygotes in F1 and F2 plants can be understood through the this diagram of this Punnett square. So this is what we understood about this monohybrid cross. So I think you are all very much clear with this cross having two stages, F1 and F2 generations, and what all are the genotypes and phenotypes. So let us move forward with the other parts. Now, here, the possible gametes are written on two sides, usually on top row and the left column. So we have seen here in the top row, left column one is written, on the right column another one is written. So the parental TT here, we are taking this as a male, and this is the, in the first filial, this is the male and this is the female. So first we told that first we are just doing the cross. I'll tell you that which one is male, which one is female at the end. So this one is male and this one is female. Okay. So similarly, this in the first F1 generation, this dominant male part is tall plant is male and the recessive was female. The gametes produced by them in the F1 progeny are TT. So again in F1 progeny, the TT are self-pollinated. So there the symbols of female and male are used to denote the female eggs and the male in the F1 generation respectively. So you are denoting the male with this one symbol and the female with this one. Okay. So like this you are denoting the male and the female. And in the F1 the genotype is TT. When self-pollinated, it produces gamete of the genotype T and T in equal proportion. So when the fertilization is again taking place, So T have the chance of 50% to pollinate the eggs, and genotype T as well as the genotype T also has the 50% chance of pollinating the eggs. Okay. So this genotype and this T genotype, they all have 50% chances of pollinating the eggs, right? So as a result of random fertilization, the resultant zygotes like TT, TT and TT are formed. So we have seen in the F2 generation, their genotype ratio is 1:2:2:1. So that is the genotype ratio which we have seen in the second filial generation. So now we will move forward with the next part in the Punnett square. So as after random fertilization, the resultant zygotes of the genotypes were TT, TT and TT. So from the Punnett square, it is easily seen that 1/4 of the random fertilizations led to TT, that is homozygous tall plants. Okay. Now half of that lead to TT, that is heterozygous, and 1/4 led to TT. So though the F1 have a genotype of TT, so F1 generation all are having a genotype of TT. So they are heterozygous, but genotype is heterozygous, but they phenotypically look tall, all of them. But in the F2 generation, what we see, 3/4 of the plants are tall, whereas some of them are TT while others are TT. Okay. So all are tall, but some are genotypically homozygous and some are heterozygous. Okay. And externally it is not possible to distinguish that which all are heterozygous tall, which are homozygous tall. So we all are seeing that 3/4 of that full lot are they are all tall, and only 1/4 is dwarf. Okay. So, hence with the genotype, a pair of TT, only one character T expressed is tall. That is, we can say it is dominating over the allele T or the dwarf character. So, small t is the dwarf character or it is the recessive character, and the capital T is the dominating character, that is the tall character. So, it is clear to all of you, right? Now, it is thus due to the dominance of one character over the other that all the F2 are tall, though the genotype is TT. Now, in the Punnett square, we have seen that in F2, 3/4 of the plants are tall, though genotypically half are TT and only 1/4 are TT. So, this leads to the phenotypic ratio of 3:1. So out of this 3, the genotype is 1/4 is TT and half is TT. Okay. So, and we can say that the rest half, 1/4 is TT. So 3:1 is the ratio, and but the genotype ratio is 1:2:2:1, 1:2:1. So this is the genotype ratio. So this is TT is to TT is to TT. So this is how the genotype is divided, and this is how the phenotype ratio is divided. So is it clear to everyone now? It is a bit difficult, but if you understand this first part for the dihybrid cross also, it will be very easy for you to understand that. Now, the 1/4, the half and the 1/4 ratio of TT, TT and TT are mathematically condensable to the form of binomial expression AX plus BY square. Okay, so the binomial expression is taken as that the gametes bearing T and T are equal frequency of half, and the expression will be expanded as given below. So this is the expanded expression, and to form a binomial expression of AX plus BY square is this, which is expanded in this form, right? Now, Mendel self-pollinated the F2 plants and found that F2 plants continue to degenerate the dwarf plants in F3, F4 generations as well. So, the recessive character which was not showing its phenotype in F1 generation started showing after F2 and F3 generations. By simply looking at the phenotype of the dominant trait, it is not at all possible to know that the genotypic composition of those plants. Okay. So hence, for example, when a tall plant from F1 generation or F2 generation composition cannot be predicted that whether it is genotypically homozygous or heterozygous, Therefore, to determine the genotype of the tall plant, at F2 generation, Mendel crossed the tall plant of F2 with a dwarf plant. So, what did he do? In F2 generation, he crossed a tall plant with a dwarf plant, right? So, this cross of a tall plant with a dwarf plant is called the test cross. Okay, so this is the test cross. Now, in a typical test cross, an organism P plants here shows a dominant phenotype and whose genotype is to be determined. So, we are seeing an observable phenotype, but we are going to take out or find out the genotype of the plant and is crossed with a recessive parent instead of self-crossing. So, instead of self-crossing between two tall plants, he took a tall and a dwarf plant and crossed. The progenies of such cross can easily be analyzed to predict the genotype of the test organism. So, what he did, he took a violet color flower that is denoted as W dominant over a white color flower which is recessive. So, using Punnett square now, your homework will be you are using the Punnett square. You are crossing this dominant violet flower, with a white flower which is recessive, right? So what ratio did you get? This, I want your answer, what you are getting. So this we will be discussing in the next class. Use the genotypes of this cross. Can you give a general definition of test cross? So this monohybrid cross, we have done the Punnett square today in details. So this is what you try to do at home. What we will do in the next class, tomorrow itself we will be doing this one. So this is the Punnett square. Like this we will do, and based on these observations of monohybrid cross only, Mendel's, what did he do? Today's principles, laws of inheritance, that is the first law of dominance and the second law of segregation, they came into existence. So this too we will be studying in the next day's class, and these are very important, which will lead us to the incomplete dominance, and then only we can move on to the next topic, that is codominance. So these are very important parts which we are going to deal in the next class. So for that you need to subscribe to my channel, stay tuned, stay updated with all the lessons which I am posting, and please don't skip my lessons in the middle, because it is throughout the lesson I am teaching all the important facts and I am giving you important notes, definitions, and also don't skip, stay tuned throughout the lesson. And please don't forget to share and subscribe to my channel. If you are having any queries, any doubts, please do write in my comment box as I can get back to you in the next episode. So stay tuned, stay updated. Thanks a lot for watching my video, and please do go through all the episodes.